Now, welcome to one of the first video recording for the first unit in your National 5 Biology. There are 3 units in total, and this is the first key area in Unit 1. And that first key area is called cell structure. It's cell structure and function. So we're going to look at cell structure of plants, animal, fungi and bacterial cells, just the basic structures that are in those cells and the jobs that the structures do. It will take you a bit of time to get used to using these videos, but the beauty of them is that you'll be able to stop them and pause them and make your own notes on the information that I'm going to give you. So the first piece of information we're going to have a look at is looking at an animal cell. Now, we've already done this, I think it was in first year, but we're going to go over the basic parts of an animal cell and the function of those parts. Now, the first part, working from the outside in, is this here, the cell membrane. And down here you'll see that the cell membrane controls what enters and leaves the cell, and we'll look at that in more detail when we come on to have a look at the actual structure of the cell membrane. But basically, if you think of it just now as controlling substances in and out. Then next you've got this here called the cytoplasm. Now, the cytoplasm is the area in the cell where chemical reactions happen. And then lastly, you've got the nucleus. And the nucleus has got in it the chromosomes in the cell, and it controls the cell. It contains all the genetic information, and it's involved in controlling the formation of proteins and such like. And we'll look at that in more detail as we go on. So we can have a wee look at just a picture of an animal cell under the microscope. And here's one here. These are cheek cells, and they're stained with a dye called methylene blue. And you can just make out the cytoplasm, the cell membrane, and the nucleus here that's stained in that darker colour. So if we move on, and we have a wee look now at the structure of a plant cell. Now, plant cells have got the same parts as an animal cell. Plant cells have a nucleus, have cytoplasm, and they have a cell membrane. But there's additional structures that we're going to have a look at. There's this structure here, which is shaded in blue in this here, and that is, whoops-a-daisy, this is my new pen, and I'm not sure how to, I don't want to do that. I'll just sort that. And I'll change my pen again. So we've got this structure here, which is shaded in the blue, and that's called the vacuole. It contains cell sap, it's got sugars and salts in it, and it helps to maintain the structure of the cell, or gives it some support within there as it kind of fills up with the sap. It helps to hold the cell in a shape that gives it some support. Now here we've got a number of structures here. They're called chloroplasts, and chloroplasts contain chlorophyll, and that chlorophyll is used in absorbing sunlight for photosynthesis. And again, we look at that in more detail further on in the course. And then lastly, we've got here the cell wall. Now, the cell wall is made up of a substance which is called cellulose, and that's a strong carbohydrate, and it is able to give the cell some support. It helps to keep the plant cell with some sort of rigidity, or gives it support, is how we usually describe it. Because you have to remember that plant cells aren't stuck on skeletons or anything like that, so it helps to hold the plant upright. Now here we've got some cells underneath the microscope. Over at this side we've got, oh, it's just moved away, sorry. We've got a substance here, or a plant cell called Elodea. That's a pondweed, and this here, if you have a wee look here, you can see, I'm not very good at drawing it, but along here is the cell wall and the cell membrane. Let me just take it up in a wee bit more detail. There's the cell wall. Oh, it's quite a thick pen. and the cell membrane is there, and then as well as that you can see these structures that are here that are green in colour, and these are the chloroplasts, and you'll see there's lots of them inside here. Now the other one, the other cell that's there, is called cobomba, and cobomba is a water plant as well, and we use that in the laboratory. You've probably used it already. Now in this case here it's been dyed with a methylene blue dye, so you can make out the cell wall structure, and then within that the cell membrane, and you can make out the nucleus there as well. So it's quite handy just to see what these structures look like. Now sometimes when we're looking at this unit of work, we get questions which are around calculating cell sizes underneath a microscope. And here's just one of these example questions that can sometimes arise. And the question is here, it's telling you that the field of view, that is when you're looking down the microscope, you see this field of view running across here. That's from one end of where you're looking through the lens to the other end. You're looking at a number of cells that are contained within that field of view. And it tells you that the width of the field of view from, oh wait, and I'll just get my pen up and working, hopefully. I'll go back to where I want to be, which is on, my slide is moved on. Just have to bear with me while I get used to using this new system that I've got here. If we have a look here, here's a field of view. I don't want that to draw on zoom, sorry about this. Take that back to the size that I want, and I'll try and get this pen to work again. Here's the field of view here, from this end here to this other end. Oh no, please don't do that. Sorry about this. Trying again. So we've got this field of view that is here at 0.5 millimeters across the way, and you can see that red line that is there. And the question asks us to calculate the width of one cell. It says here a wee clue is to count the number of cells from one side to the other across the line shown and estimate the cell length. So what you would do is you would count the number of cells in the field of view that you've been given here. And we can just make out there's a cell here, 1, and there's a little bit of a cell over here. So we can count that really as one cell, the total of these two pieces that are there. So one cell, 2 cells, 3, 4, 5, 6, 7, 8. So there are 8 cells across this field of view, which is 0.5 millimeters. To work out what the width of one cell is, you would just divide 0.5 divided by 8, and that will give you the answer 0.0625 millimeters. So each individual cell is 0.0625 millimeters in length. Now here's an actual exam question. Now this exam question says that the diagram below shows onion cells observed under a microscope at a magnification of times 100. Shows us the field of view here, this circle, and then inside there are these onion cells. Now it tells us that that field of view is 300 micrometers across. Now you may or may not have come across the term micrometer. It's a measurement, and a micrometer, if we look at a millimeter, which you know, 1 millimeter equals 1,000 micrometers. And sometimes you see micrometers represented by a sort of odd looking U. That's the micrometer. You don't usually need to know the size of this at National 5, but you may come across the term just so you know what it's about. But you don't need to worry about it for this question here, because you're not needing to change micrometers to millimeters or anything like that. But the question says the diameter of the field of view is 300 micrometers. the average width of each cell in micrometers is, and you're given a choice, A, B, C or D. But you don't have to guess the answer to this because you can work it out quite easily. You can look at your field of view and count how many cells are there across the field of view. Now here there's 1, 2, 3, 4, 5, 6, 7, 8 cells. So if it's 300 micrometers across, and there are 8 cells, 300 divided by 8 gives you the answer 37.5 micrometers. And that's how you would work it out. And you may come across examples of questions like this in the examination. Now we're going to have a look in a wee bit more detail at the actual structure of a cell, sometimes known as the ultrastructure. And so far we've looked at pictures of cells under the light microscope, and it can magnify things up to about 400 times, or if you've got a very expensive one, 1,000 times. But to look at more detail inside cells, they use something called an electron microscope, and that uses a beam of electrons instead of light, and it can magnify things up to about 2 million times larger than they actually are, and it allows us to see smaller structures inside cells. So example of that is looking at these pollen grains that are over here under the light microscope, and then when you look at them under the electron microscope, and you'll see that you can see much more detail in the structures of the outside of the pollen grains and see the differences that there are there in the structure. Also, if we have a wee look at, say, an animal cell in the electron microscope, you can see there's lots of other little structures inside here. The same with a plant cell as well, so we just have a wee look here. Here's a plant cell in the electron microscope, and you can see how you can see much more detail. There's a nucleus with another area here called the nucleolus in it. So we're going to have a look at some of the structures that you need to know in more detail inside the cell. In other words, the cell ultrastructure. We call these organelles inside the cell. And one organelle that you may or may not have heard of is this one here called the mitochondria. Now, mitochondria are involved in aerobic respiration in the cell. So they're the place where one of the stages of respiration takes place, called aerobic. That is the part which needs oxygen. And that's your mitochondria. We'll look at these in more detail. Not very good at writing on this system here yet, so hopefully you can make that out. And the other organelle that you need to know about is this one here called ribosomes. And ribosomes are involved in protein synthesis. That is, in making proteins in the cell. And you can find them sort of freely about the cell, or sometimes you find them attached to another structure which has got the name endoplasmic reticulum. You don't need to know that, but the ribosomes can be attached to that, and it's called rough endoplasmic reticulum when that happens. So these are the two other structures that you need to know about found inside a cell, and these are found inside mitochondria, and ribosomes are found inside plant cells as well. So we're going to move on and have a wee look in more detail at fungal cells. Now, you know fungi already. You know mushrooms and toadstools and molds. The one that we're really interested in is, or I'm saying it, the one that we're really interested in, well, the one that we talk about quite a lot are yeast cells. They're unicellular fungi. Now, we look at yeast actually quite a lot because we use it in a number of experiments to illustrate some processes that take place in the cell, mainly respiration. When you look at yeast underneath a microscope, here's a yeast cell here and a yeast cell here, and these are lots and lots of little yeast cells that are here, and you'll see there's little buds just coming off the cell. That's how yeast cells reproduce. They just produce wee buds, and then these wee buds break off and there's a new yeast cell. Now, we have a wee look at the typical structure of a yeast cell, hoping to find that on this slide here next. There's a cell wall, cell membrane, a nucleus, a vacuole, and there's cytoplasm. It doesn't have chloroplasts like a plant cell, and it also actually has ribosomes, but they're not there on this picture here. These wee dots that are here are representing mitochondria. So it has mitochondria and ribosomes too, and it has a cell wall similar to a plant cell wall. Now, yeast is used in a number of industries. It's used in bread making, it's used in beer making, wine making, whiskey making. It's also used in making alternative fuels as well. So that's a yeast cell. We're going to move on and have a wee look at bacterial cells and have a look at their structure. And this is just showing you the size of bacteria cells and how small they are compared to plant and animal cells. Whoopsie daisy. So it's just showing you the relative sizes that are here. And here's your bacterial cell here, down at 1 micrometer. Remember I said before that's 1,000th of a millimeter, so here's a millimeter up here. Anyway, let's have a wee look at the structure of a typical bacterial cell. This is a bacterial cell underneath the microscope up here, and they have some structures that we've seen already in plants and animal cells. They have ribosomes, they have cytoplasm, they've got a cell membrane, they have a cell wall, but that cell wall is made up of different material than a plant cell wall. You'll see this tail here, that's called a flagella, and it helps, some bacteria have these, and it helps the bacterial cell move about. When we have a wee look at our bacterial cell in a wee bit more detail, it says here, I've said here that there are some similar substance structures and the absences of some other cell organelles. It has a cell wall, and the chemical structure is different from a plant cell. You don't need to know what the cell wall is made up of, but it's a different structure, it's substance, it's not cellulose. Now when we have a wee look in more detail at the DNA that's in a bacterial cell, there is a difference in a bacterial cell in that it doesn't have what we would call a proper nucleus. The DNA is found in this circular chromosome that's here. So it's a kind of main ring or coil of DNA that's there. There are also these other smaller rings that are here, and these are called plasmids, and they're important later on when we come to have a look at genetic engineering, and we have a look at that in a wee bit more detail. So these are the structures that you need to know about. I think also on this diagram here, yeah, labelled up there, there's the flagellum is labelled up here as well. And you'll see here that there are 3 flagellae. A singular one is flagellum. There's 3 of them, so some bacterial cells can have more than one. I've made a wee note on this next slide here just to emphasise the cell wall structure, that the cell walls that surround the plant cells are made up of cellulose, but the cell walls that surround the bacterial cells and the fungal cells are not made of cellulose. So that's important to know that. Now we're going to have a wee look at reproduction of yeast and bacterial cells, and this sort of thing comes up in questions. Here we've got this situation here. It says if the conditions are right, yeast and bacterial cells can divide to make 2 cells once every 30 minutes. This means that the number of cells can double every 30 minutes. So that's pretty quick. So we're going to be having a look at this sort of question here. It's saying 1 yeast cell placed in a sugar solution, it divides to form 2 cells in 30 minutes. And the question asks, how many yeast cells will there be after 12 hours? Now, what we're talking about here is that this is going to double every 30 minutes. And sometimes when people answer this question, what they do is they just would think, oh well, 12 times 30, that will work it out. But we need to have a wee think about this, and how I do that is, if I draw this out, it helps hopefully to see what is happening. Here is one cell. I'll just try and draw that out. Hopefully this won't start to magnify. Here's one cell, and after 30 minutes we end up with 2 cells. Then another 30 minutes time, what's going to happen is each of these cells are going to divide. So that will give us 4 cells after an hour. 2 after 30 minutes, 4 cells in an hour. Another 30 minutes later, each of these 4 cells are going to divide. We'll draw them out. So, and therefore we end up with 1, 2, 3, 4, 5, 6, 7, 8 cells. Another 30 minutes later. And then another 30 minutes later, in other words, at the 2 hour mark, these 8 cells, each of them is going to double, and therefore we'll end up with 16 cells after 2 hours. What's happened is that it keeps doubling every single time. Now if we have a look on the next slide here, I've got that put together to let you see what actually happens. So here, the question that was there way back in the beginning was, where is it? How many yeast cells will there be after 12 hours? Now because it's 30 minutes, there's 2 hours in every, there's 60 minutes as we know in every hour, so that's 2 lots of 30 minutes. So we can double 12 hours to make it 24, and we can write out, you'll see here, I've got at the first stage, in other words the first 30 minutes, there'll be 2 cells. Then the next 30 minutes there'll be 4, then the next 8, 16, 32, 64, 128, 256. So you'll see it's doubling every single time. Now it's doubling, it's actually going to double 24 times because we're talking about 12 hours here. And at the end of the 24, in other words at the 12 hour mark, the answer is, look at this, 16,777,216 yeast cells. So a lot of yeast cells after the 12 hours. I'm sorry this keeps jumping about. We'll get used to it in the further videos, and it hopefully won't be quite so jumpy. It's just taking me a bit of time to get used to this new system that I've got. So that is how you would work out. You wouldn't normally get a 12-hour question usually, but you will get questions where it asks you to work out the number of cells after a certain length of time, and that's how you would do it. You would need to think about how it doubles and how many times it's doubling, and we'll see if we can find some example questions like that. Lastly, up on here, we've not really talked about the structure of a virus. Viruses, they're not actually made up of cells. They don't have a cell membrane, a cytoplasm or a nucleus, and there's some debate whether they're actually living organisms or not, but they have got a protein coat and they contain some genetic information inside, but a virus, it can't reproduce by itself. It's got to infect other cells, so we kind of talk about them not really truly being alive themselves, and that's why we've not really talked a lot about their structure. But you'll see there's some wee DNA in this odd virus that's up here that I've got a wee picture of, and a wee protein coat. But viruses come in all sorts of shapes and sizes and as well. So that's the end of Key Area 1. It's quite a long video to be watching and making notes on, and I'm sorry, it seems to be there's a bit of a hiss in the background here as well, but hopefully you'll have been able to have made some notes. Thank you.